An overview of Cyanophyta. Now, cyanophytes are also called mixophytes. This is a very primitive group of organisms, and these are bearing similarity with both algae as well as bacteria. Hello friends, I am Ayantika, and in this video I will be discussing various aspects about the cyanophytes, and this video there will be two parts of this video, and I have also made videos regarding bacteria and algae. So the description in the description box I will be putting the link to all these videos. So let's start the video. Before we start the video, uh, there will be a quiz. Red color of the Red Sea is due to, and there are four options: iron ore in the seabed, or this Trichodesmium erythraeum, red colored aeoline deposits from nearby land masses, the red colored dye that entered the Red Sea as effluents. So I will be providing the answer at the end of the video. However, if you know the answer, you can write it in the comment section. So let's start the video. The cyanophytes or the mixophytes, these are very primitive organisms, and they have an autotrophic mode of nutrition. That is, they photosynthesize their own food. So at the same time, they have two very important ecological role, because in the primitive world, the atmosphere didn't have much oxygen. So these mixophytes or the cyanophytes, the blue-green algae, or we can say the these category of gram-negative bacteria, they are the sole producer of oxygen in the primitive environment. The ancient rusting of earth is due to this group of organisms, because apart from photosynthesizing, they produced oxygen that was quite deficient in the earth of that time, and due to this reason, this excess amount of oxygen produced, the iron oxides in the ocean, the iron ore in the ocean, they were oxidized, and this event was called rusting of earth. And this rusting of earth is mainly contributed by the mixophytes or the cyanophytes. Apart from this oxygen production, their own autotrophic mutation, they are also fixers of atmospheric nitrogen. They have this mechanism, and this nitrogen fixing mechanism of the cyanophytes are utilized by many other organisms on whose roots these cyanophytes can be found. We will be discussing it later in the video. These cyanophytes, they are prokaryotic, which means that they do not have a true nucleus. Their nuclear material is just coiled DNA with some RNA material, but unlike the eukaryotic organisms, it does not have a well-defined nucleus surrounded by a nuclear membrane. It's just coil of DNA and some extra nuclear DNA like materials, and some RNA, and also the 70S ribosomes, just like the normal bacteria have. All the eukaryotic organisms, they have 80S ribosomes, but this cyanophytes, they have 70S ribosomes, just like other bacteria. And just these cyanophytes, one of the main characteristic that makes it closer to the bacteria than to the algae are that it does not have any flagellated cell even in the reproductive stages. So this is the lack of flagella brings brings it very much closer to the bacteria, apart from another important factor, that their cell wall is very similar to the cell wall of gram-negative bacteria. And in fact, this is also a gram-negative bacteria um, because of the staining capacity. And then but it also shows movement. It does not have flagella, but it shows movement in the form of very glidey or jerky type of movement. The photosynthetic apparatus is mainly limited to the folds of the plasma membrane. It does not have as it is a prokaryotic organism, it does not have any membrane bounded organism. So the proper chloroplast in the chromatophore or is not is actually absent. Whatever photosynthetic mechanism is located just in the folds of the plasma membrane in the form of chromatophores. It does not have the thylakoid-like structures very common to the eukaryotic photosynthetic apparatus. However, it has some pigments like chlorophyll A. Chlorophyll A is common to all green plants and absent in generally bacteria. So this presence of chlorophyll A brings it again closer to the algae rather than to the bacteria. Other photosynthetic pigments that are present are the phycobilins, that is the phycocyanin, phycoerythrin, beta carotene, and xanthophylls. Xanthophylls it includes mixoxanthin and mixoxanthophil, which is characteristic of this Cyanophytes. So four type of pigments are present in the photosynthetic apparatus, and apart from that, the main storage material is mixophycean starch or cyanophycins. The mixophycean starch is a carbohydrate and cyanophycin is a protein storage product. The the cyanobacteria, it has a outer mucilaginous matrix of protein surrounding the cell wall. Cell wall is of gram-negative bacteria type, and inner to the cell wall is the plasma membrane. And plasma membrane has many folds which act as photosynthetic apparatus, and the cytoplasm is actually divided into two parts. The outer richly colored ectoplasm or the chromoplasm, and the inner colorless centroplasm, which contains the genetic material. The centroplasm, it is generally transparent and has only genetic material, and the external the chromoplasm, apart from the lamella of the plasma membrane, which acts as a chromatophore, also the external cytoplasm contain many other inclusions in the form of the granules of ribosomes, storage granules, and if it is living in a residing in a very high nutrient atmosphere, especially high amount of phosphate containing atmosphere, then polyphosphate granules are also found. Polyphosphate granules are generally found in very the polyphosphate granules are present in large amount, mainly when the nutrition condition is high. Otherwise, there are polyphosphate granules, but not in high amount, as it is a prokaryotic organism. So the vacuoles, they are not true vacuoles. They are just gas filled structures that do not have membrane, but instead the membranous part is replaced by proteinaceous cover. And these vacuoles, pseudo vacuoles, are helpful in maintaining the buoyancy during the photosynthesis. And what happens is that when loss of uh, loss of the photo assimilates, there is very low amount of sugar in the body of the bacteria. uh, it comes up the comes up from the bottom of the water body, and it photosynthesizes. And as it photosynthesizes in the upper layer of the water, it accumulates the starch, the Cyanophycean starch. And once the threshold of Cyanophycean starch is met, the bacteria, cyanobacteria, it loses gas from the pseudo vacuole, and it settles down to the bottom of the water body in which it is living. So by this help of pseudo vacuole, they become buoyant, or they settle down. They generally become buoyant during the early period of the morning, that is from 12 to 3, that it is a maximum photosynthetic time. So if you want to collect cyanobacteria, it is best to collect cyanobacteria from when the time is from 12 p.m. to 3 p.m. So the pseudo vacuoles functions for the buoyancy. Next is the extremophilic property. All the cyanobacteria, they are extremophiles, because which means that they can survive under very high as well as very low temperature in acidic as well as alkaline environment, in water, as well as it can survive in highly arid condition also. This is because of a range of factors, especially this cyanobacteria, they have a mucilaginous outer covering outside the cell wall. This mucilage protects us from desiccation, and also from predation by other animals, because due to the copious secretion of mucilage, they are not that much palatable to other organisms. And apart from this protection against grazing, this mucilage also helps in tiding with short periods of desiccation. For long period of desiccation, it produces heterosis, that I'll be discussing in the next slide. And this during the extreme temperature of high temperature or high aridity, it produces perinating structures called echinites, or heterocysts, which are resting. And this akinete is not actually resting, but more is the heterocyst that we'll be discussing in the next slide. Now, this apart from the gelatinous outer sheath, it also have certain proteins which are very compact in the molecules of the protoplast. And due to this gelatinous sheath and compactness of protein and heterocyst, they are able to tide over temperature extreme, pH extreme, as well as aridity extreme. And this cyanobacteria, they were actually in earlier time, it were included in inside the algae, because of the chlorophyll A content in it, and also autotrophic mode of nutrition. So it had orders like Chlorococcalis, Oscillatorialis, Nostocalis, Cytonematalis, and Revularialis. So I will be discussing the various orders in detail in a few videos, in the few upcoming videos. So I'm not going to discuss all these orders in detail now. I'll be discussing it in few next videos. So this cyanobacteria, they are more they have a cosmopolitan distribution. They are found both in aquatic as well as terrestrial environment. The aquatic environment is mostly freshwater. However, we can find many examples of cyanobacteria living in marine environment. In fact, all the marine there we can get many examples of marine blooms that are caused by the cyanobacteria. And in while terrestrial in aquatic environment also, apart from being a planktonic or benthic form depending upon the time of the day, that is during the early period of the day when there is intense sunlight, it rises up and is present in the pelagic layer as plankton. But as the intensity of light decreases and it has fulfilled this quota of photosynthetic accumulation in the body, it sinks to the bottom. The bottom of the water body is rich in phosphates, nitrogenous substances. So they also absorb these minerals from the bottom. So in any environment, aquatic environment, they are also found growing inside the shells of aquatic organisms also. And in terrestrial environment, they are found growing in the inside damp soil, or on the root nodules, or even certain organisms like they have a symbiotic association with the cycas root inside the thallus of bryophyte anthoceros, in the plant body of azolla, the water fern. And also it has symbiotic relation with the lichen in the form of lichens, where the fungi and green algae and the cyanobacteria, they form these three form of symbiotic association. And also they form a symbiotic association with the haptophytes. Now we know that this haptophytes, because of their hard exoskeleton, they are the most abundant microfossil found in marine environments. And they are deposited. They are the chief source of chalk deposits. Chalk deposits are quite found in very commonly in the in and around the United Kingdom. And these haptophytes, they act as food material for aquatic micro invertebrates like the shrimp larva, the uh, the small organisms. So I will be discussing very detailed about the symbiotic relation of the cyanobacteria in the next part of this video. Uh, and I'll be talking about blooms now. The blooms, the blooms are formed only when the amount of cyanobacteria crosses a certain threshold. Actually, they by quorum sensing, when they know that they have abundant number of cyanobacteria around them, they start reproducing at a very fast rate, and blooms are formed. Blooms are sudden increase in the number of bacteria or cyanobacteria. And but the aquatic blooms, they are formed both by cyanobacteria as well as algae. The algal blooms are not toxic, but the cyanobacterial blooms are very toxic. Even when the cyanobacterial blooms are not present, then also certain amount of toxins are also released by the cyanobacteria. These toxins, they are not released by the living cyanobacteria, but by the breakdown of walls of the dead cyanobacteria, that these toxins are released in the environment. And these blooms are generally found throughout the season, throughout the year in the tropical region, but in the temperate region they are found mostly during the warmer months, that is the summer season. Now I come to the end of the video, part 1 of this video. Well, there is a question, the quiz. The red color of Red Sea is due to the red color of Red Sea, the color of any water body can be attributed to different causes. It can be due to the it can depend upon the ores of the bed material, because the ores can form oxides or hydroxides or whatever, and depending upon the type of metallic oxides or hydroxides or metallic ion that is present in the bed material, the ionic form or the oxide or hydroxide form can dissolve and attribute color to in the water body. The color of water body can also be due to the effluent dyes that enter the water body, because we know that the organic matter, though organic matter also decompose produces colored, they are colored in the water body, but dyes are certain, the color produced by dyes are more or less permanent, because dyes are they decompose at very slow rate, much slower rate than the organic matter. Water body can be also colored due to any dye effluent that is entering the water body. Also, the color can be temporarily added by the aeolian deposits of the landmass that is surrounding any water body. But here, the color of the Red Sea is mainly attributed due to this blue-green algae or the cyanobacteria Trichodesmium erythraeum. Trichodesmium erythraeum, it produces red pigment, and the color of the Red Sea is only and only due to this Trichodesmium erythraeum. And with this, I come to the end of the first part of this video. In the next part of the video, I will be discussing about the thallus structure and about the reproductive procedures and the economic importance of cyanobacteria. And I will be discussing each and every cyanobacteria from all the orders that are in the syllabus for MSc and BSc. These videos will be soon following this video, and so thanks for watching the video. And I have put links to the algae or lichen and in the description box. So if you want me to make any video, or if you are not clear, you want me to make more a detailed video on anything, please write in the comment section, and please share the video with your friends if you like it. Subscribe this video to get updates about the latest videos I make. Thank you.